Welcome to Pharmacron. In today's lecture, we're laying down the essential foundation for pharmacology and medical microbiology, the principles of antibiotic therapy. So, what is an antibiotic? Simply put, antibiotics are agents that selectively target and kill bacteria, what we call bactericidal, or inhibit their growth, known as bacteriostatic, all without causing significant systemic toxicity to the human host. Originally, the term referred strictly to naturally derived substances produced by microorganisms, like penicillin isolated from penicillium fungi. Today, however, our definition encompasses semi-synthetic derivatives, as well as fully synthetic compounds, like fluoroquinolones and sulfonamides. Next, let's talk about the spectrum of activity. This refers to the range of different microbes against which an antibiotic agent acts. We generally break this down into two main categories. Broad-spectrum agents are effective against a wide array of organisms, targeting both gram-positive and gram-negative bacteria. Narrow-spectrum agents, on the other hand, target a smaller, specific group of microbes. For example, agents that act exclusively against gram-positive organisms.
When pharmacologists design these drugs, what are they aiming for? Let's look at the 10 qualities of an ideal antibiotic agent. First, it must be selectively toxic to the bacterial cell. It should preferably be bactericidal rather than bacteriostatic. It must exhibit a slow emergence of resistance. For targeted therapy, it should ideally have a narrow spectrum of activity to preserve normal flora. It must be completely non-toxic to the host. It should possess a long plasma half-life, allowing for convenient once-daily dosing. It needs good tissue distribution, including penetration into the cerebrospinal fluid, CSF. It should have low plasma protein binding, ensuring high active drug levels. It ought to be available in both oral and parenteral formulations. And finally, it must show minimal drug and food interactions.
Now, to kill or inhibit bacteria, we must target structures or pathways essential to their survival. Here are the 5 major target sites and the physiological rationale behind them. 1. Cell wall synthesis, the peptidoglycan layer. Bacteria maintain massive internal osmotic pressure, up to 20 atmospheres in gram-positive species. Without a rigid peptidoglycan mesh serving as an exoskeleton, water rushes into the cell, causing rapid osmotic lysis. It also maintains bacterial shape. 2. Protein synthesis. Proteins make up over 55% of a bacterial cell's dry weight. Ribosomes constantly translate mRNA into enzymes, structural units, and virulence factors. Halting protein synthesis prevents bacteria from doubling their population. 3. Nucleic acid synthesis. During replication and transcription, circular bacterial DNA must unwound, generating extreme supercoiling. Bacteria rely on DNA gyrase to introduce negative supercoils and topoisomerase 4 to untangle daughter chromosomes after cell division. Inhibiting these enzymes halts genome replication. 4. Metabolic pathways, folate pathway. Bacteria rely on tetrahydrofolate, THF, to synthesize purines, pyrimidines, and essential amino acids. Unlike humans who ingest folate through diet, bacteria cannot absorb exogenous folate across their cell wall. They must build it from scratch using PABA. 5. Cell membrane function. The plasma membrane controls ion gradients and nutrient transport. Disrupting this lipid membrane leads to rapid leakage of essential ions and loss of membrane potential.
Finally, how do we organize all these drugs? We classify antibiotics in 2 primary ways: by chemical structure, penicillins, cephalosporins, fluoroquinolones, tetracyclines, macrolides, aminoglycosides, chloramphenicol, antimetabolites, and lincosamides. By mechanism of action, or MOA, cell wall synthesis inhibitors, such as beta-lactams and vancomycin, nucleic acid synthesis inhibitors, including folate synthesis inhibitors, DNA gyrase inhibitors, and RNA polymerase inhibitors, protein synthesis inhibitors, targeting either the 50S or 30S ribosomal subunits, and drugs acting directly on the cell membrane. Catch you in our next video as we unravel each class of antibiotics step by step.
exploring their mechanisms of action, structure-activity relationships, clinical indications, contraindications, and high-yield exam questions, right here at Pharmacron. Don't forget to like and subscribe.